This presentation demonstrates workflows for the stable isotope labeling of protein digests and synthetic peptides employing oxygen-18-enriched water. First, in the protease-catalyzed PALeO time course experiment, incubate substrate and protease in the presence of 50% oxygen-18-enriched water to incorporate oxygen-18 atoms. Then, for proteases capable of double labeling, repeat incubation of the post-digestion cleavage products with protease of interest in the presence of 50% oxygen-18-enriched water. The ALeO workflow performs the acid-catalyzed incorporation of oxygen-18 atoms into functional groups. At timed intervals, spot aliquots of the reaction mixtures with matrix on moldy target plates. Upon mass spectrometric data acquisition, generate spectral time plots of the peptide cleavage reactions as well as oxygen-18 incorporation plots of individual peptide species. Labeled cleavage products are selected for MS-MS-based sequence identification. Ultimately, these workflows offer versatile tools for quantitative and qualitative proteomics studies.
This method can help answer key questions in the protease research field, such as identifying rate-limiting steps and reaction intermediates in complex proteolytic pathways. Proteolytic signatures identified by the PALeO time course provide important facts regarding substrate specificity. In addition, the time-resolved data also yields kinetic information. Diana Klingler, a postdoc from my laboratory, will now demonstrate the procedure.
The PALeO time course workflow monitors dynamics of proteolytic cleavage reactions based on protease-catalyzed oxygen-18 incorporation. For the 20 microliter reaction, add the sample peptides to the protease reaction buffer containing oxygen-18 enriched.